you, ma'am. Thank you so much. Okay. Here I am to present my research topic, that is the first report of Anthocyanin as a neutralizer against Takumi, which is a trade name, and the chemical name is Flubendiamide 20% WDG, that is water dissolvable granules, and induced toxicity in Labeo rohita. And here Labeo rohita is a common edible fish which is abundantly found in paddy fish ecosystem. And I would like to say that this was a part of my PhD, and the research was carried out in Vidhan Chandra Krishi Vishwavidyalaya, and presently I am working as an assistant professor in CV Raman Global University Bhubaneswar. I am recently joined in this university. So let us talk about, ma'am, please next slide. Okay, thank you. So I would like to describe the background of this research. Actually, paddy fish culture is an extensively being used practice for past many years because of the maintenance of energy and land resource. Because in limited land resource, we can use the protein major protein food, that is the fish and rice also. So but due to insufficient knowledge, some farmers use some agrochemicals in huge amounts, and these agrochemicals generally create some ecological hazards, and these create the ecological hazards in non-target organisms which reside in paddy cum fish ecosystem. And as we know that the agrochemicals include mostly pesticides, including insecticides, fungicides, and these chemicals are also used to control the different diseases of the plants, and especially in rice plants when there are stem borer and some pod borers are there. So to prevent all these insects, we use some chemicals which are insecticides. So if we use these insecticides in huge amount, this will create an adverse effect in non-target organisms which are residing in the paddy cum fish ecosystem, and the surface runoff enter into the aquatic system and induce the toxicity in these non-target organisms, especially in the soft tissues. Very much toxicity is being observed. So soft tissues include the kidney, heart, liver, muscles, spleen, all these tissues. So in this study, we have actually observed the toxicology variations in biochemical, hematological, histological, then protein profile, and some genetic level also in a long-term exposure. Ma'am, please next slide. Okay. In this slide, I would like to say something about the insecticide, which is the Flubendiamide 20% WDG. It is a contact insecticide with muscle contraction and stomach action. When the insect comes in contact with this insecticide, which is being applied to the rice plants, this insecticide induce the toxicity with contact, and the muscle of the stomach is generally hampered. And this is found very effective against the stem borers, fruit borers, jacids, diamond back moths. These are all insect pests of rice plants. And if this is a synthetic petrochemical pesticide with a rhinoid class, and this have acts as a receptors in the insect muscles, which receives some signals for this biochemical insecticide to capture these receptors, and then it induces the toxicity in that insect. And this is being formulated and supplied by Tata Rallis India. It is a pesticide company. Basically, this insecticide was purchased from Tata Rallis India company, and it is mainly used for the crops like cotton, rice, and pigeon pea, cabbage, etc. And here I have also put the molecular diagram of this insecticide. It is mostly we are seeing that cyclic cycles are there because of phenolic benzene rings are there. So it is a aromatic insecticide and induces very much high toxicity because it is not easily biodegradable, and when it is metabolized, the metabolites are intermediate metabolites are have long half life and induce very high toxicity in the soft tissues. These get accumulated also. So next, the mechanism of the action of this insecticide, if we talk about, then it will impart, ma'am, next slide. When it is orally ingested, it exhibits larvicidal activity also. Not in that means not in adult insects also, but it induces toxicity in larval stage, and it targets and disrupts the calcium ion balance in the pests, and also rapid cessation of the feeding, and finally the pest gets deteriorated or die. Next one. When it induces toxicity, so here are some references of papers I have put. So flubendiamide is not easily degraded, which I have just discussed, that due to its long half-life period of this intermediate metabolites, it causes high toxicity in the non-target organisms when these are ingested in the paddicum fish ecosystem, and long-term exposure causes the toxicity at the tissue level, very high toxicity because of the accumulation of these metabolites, and toxicity is observed mainly in liver, kidney, heart, muscles, thyroid, that is the internal glands also, and the adjacent part of the fins and eyes also. We can see that redness will be appeared in intoxicated fish. Next. Yes, some that is some ability of this Labeo rohita, which is very suitable for studying the toxicological studies, that they must tolerate to grow in shallow water, because rice feed have very depthness of water, so they are able to grow in shallow water, and they must tolerate low oxygen content, because their low oxygen content is there because of that high concentration of insecticide, which creates low oxygen content in the water, which creates high toxicity, and it also also generates the behavioral impact on these non-target organisms also. And the fish must have the capacity to grow fast and reach the market size, and they must tolerate high turbidity. That's why we are using this Labeo rohita for our toxicological studies. Next. There are few other points also. It can easily be maintained in the laboratory and does not breed in large ecosystems. This has omnivorous food habit and includes phytoplanktons, then zooplanktons, and submerged vegetation also. So in the laboratory, we feeded this fish with fish feed, and because of the omnivorous habit, it just fed very well on this fish food. And natural habitats are generally ponds. It is a freshwater fish, which natural habitats are ponds, rivers, and found abundantly in the paddy field. Next. In this slide, we can see the impact of the high concentration of pesticides in the aquatic bodies, which creates a loss of high amount of fish species in paddicum fish ecosystem. And my research is mainly oriented that how can we just rescue these non-target organisms and create more and more proteinaceous food from the paddicum fish ecosystem. So what can we do for this? Next. As we know that anthocyanins have a much better impact to cure the toxicity, because these are very strong antioxidants and work as non-mutagenic, non-tumorigenic, and also neural diseases, diabetes. These are used against cardiovascular disease, against inflammation, and anti-mutagenic and other diseases also, where carcinogens like agrochemicals can act. So anthocyanins can minimize this effect. And we have studied the impact of anthocyanins on Labeo rohita. So how can we feed these fish with anthocyanin? So for this, we have isolated the anthocyanin from the vegetable and fruits which have a high concentration of anthocyanin. Please next. So here we can see that there are few different varieties of vegetables and fruits are given in this slide, that which contain high amount of anthocyanin, which has a purple color. And we have isolated mainly anthocyanin in the from our Mondori farm from a sweet potato. The variety name is X61. Next, in next slide, it has been shown. Next. S61 is a last last slide picture shows the anthocyanin which has been isolated from S61 variety of sweet potato. And for this we have 10 samples we have just exposed to different concentrations of flubendiamide and insecticides, which has been long-term study we have used, and 10 different ppm concentrations we have applied, and LC50 was found that 2.99 ppm. This is the LC50. We have analysis through the SPSS version 19, and for Labeo rohita we have just selected 3 sublethal dose, that is the 0.498 ppm, 0.747 ppm, and 1.495 ppm. So these were selected for chronic sublethal study, and for long-term study, that was say for after 3 months, we have just sacrificed the toxicated fish and gone for the biochemical, hematological, enzymeological, and protein profiling in research. Next. So these parameters we have studied. And next, the pesticidal effect on all fish species was studied after 1 long-term study for after 90 days. And during the experimental period, the behavioral impact of the pesticide was also recorded. Next. 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 Yeah. So this is the methodology of anthocyanin, that the major coloring constituents in sweet potatoes, especially the purple fleshed varieties, have been identified as the acylated anthocyanins, and the genes IBMYB1 and IBMYB2 of MYB gene family from the purple fleshed sweet potato predominantly responsible for the anthocyanin biosynthesis. These are the 2 gene families which we have isolated, and some research papers has also been published regarding these genes, and these have been expressed in tuberous roots of tissue specific cultivar, that is the in specific manner. So in the next slide, please next. The anthocyanin biosynthetic pathway has been shown, and these genes play a very important role during the synthesis of the anthocyanin. So first is anthocyanidins are just formed. This is an inactive stages, and after the structural gene, which is the UF3GT gene, which acts on anthocyanidins and converts it into activated anthocyanins, which we require to restore the restore the control situation of the non-target organisms. Next. So how can we isolate the anthocyanin from purple fleshed sweet potato? Because there are so many processes through which anthocyanin can be isolated from the purple fleshed sweet potato. But we have finally to inject the anthocyanin, isolated anthocyanin, into the toxicated fish. If we isolate the anthocyanin from the purple fleshed sweet potato using acetone or alcohol, then it will create many more hazards in addition to the insecticides in the non-target organisms. So we have isolated the anthocyanin using the 30%, 50% and 70% solvent, that is a methanol and water. Mainly the water constituent is very high here, and the methanol constituent will evaporate. It will just volatilize, because it is an alcohol, it will volatilize. And then the supernatant was preserved in each step, and the OD was taken at 657 nanometer, and the crude anthocyanin was in powdered form, and we dissolved in just water content and injected at the dose of 5 mg per ml at an interval of 15 days for 1 month interval. And then when we observed that it is creating a very good impact on toxicological fishes, toxicated fishes, then we applied for much more, extended the application time for 90 days, and after 45 days I have created these data, and here I have just shared this data here. And next slide. When we observe the behavioral pattern of the toxicated fish in comparison to the control fish, we just observe that the toxicated fish, it is in lethargic mode, and it comes to the surface of the water, engulfs air, because there is very less oxygen content in the toxicated water. So it just comes to the surface of the water just to breathe. And bleeding occurs around the eyeball and the base of the pectoral fins also, and frequently comes to the surface of the water. And change of body color of the fish is also being observed, whereas the control fish is all shows all the normal patterns. Next. When we go for biochemical analysis of toxicity, the blood was collected from the caudal vein, and EDTA was added because of the as a non-anticoagulant, and just blood was centrifuged using 2000 rpm for 5 minutes at 4 degrees centigrade, and all the uses manual was followed. And mainly for the biochemical analysis we used next glucose, cholesterol, total protein, serum albumin, albumin by glutamine ratio, then SGOT. These are the some enzymes which are related to the toxicological studies, SGOT and SGPT, and then the creatinine, then alkaline phosphatase, cortisol, etc. These are different biochemical parameters which we studied for the toxicological aspect. Next, for hematological aspect, we just collected the blood, and immediately we transferred it into the acid tubes containing the EDTA, and followed for the hematological parameters using the user's manual. In the hematological parameters, next, RBC, WBC, packed cell volume, then hemoglobin concentration, MCV, MCH, MCHC, that is the mean corpuscular hemoglobin concentration, has been studied. Next. For enzymological studies, we studied mainly the stress-related enzymes were studied, that is the lactate dehydrogenase, malate dehydrogenase, and peroxidase. And the users' manuals were followed for the study, and the native page was done for the quality study of these enzymes, stress-related enzymes. These were all carried out according to the standard protocol. Staining purpose was also there according to the standard protocol for these enzymes. Then the protein profiling. Protein profiling of the toxicated fish showed the high amount of stress related proteins, because the toxicated fish soft tissues, that is the kidney, then liver, muscle, these showed very high amount of toxicated proteins because of that insecticide, ingested insecticide through the gills. And protein profiling was also done using the user's manual. The protocol was just followed the user's manual. And for characterization of protein, we done SDS page, and 10% polyacrylamide gel was casted, and staining was processed using Kumasin Brilliant Blue. Now come to results. What we got next. Biochemical parameters. If we see the biochemical parameters, then we can observe here that after long-term exposure, glucose level was very high in comparison to the control, and cholesterol was also very high, and these all ratios which we parameters which we studied for the toxicological studies, and after administration of anthocyanin, and the data was taken after 45 days, we see here that it is very close to the control fish. This is the main objective of this study or research, that after administration of anthocyanin, the parameters which we used during the toxicological studies was restored to the control control data. Then next, the description of the biochemical parameters. If we see that the biochemical sublethal effect of the Takumi on certain biochemical parameters of Labeo rohita, that is comes for the level 3, that is the dose 3, and the highest dose I incorporated here was the other 2 doses was sublethal doses and was very less in comparison to dose 3, and the data was not incorporated here. This table just denotes the highest, that is the dose 3 observations, that is the data. And when we administered the anthocyanin, these were all restored to some extent. It was very significant. And now coming to hematological data, here also we got the similar pattern of result, that after administration of anthocyanin, the data was very much close to the control fish. Next. Next, the enzymological parameters in intoxicated fish. Also, when we compared, next, when we compared the enzymological parameters, that is the lactate dehydrogenase, malate dehydrogenase peroxidase in different soft tissues, that is the gill, heart, liver, kidney, and muscle, the after administration of anthocyanin, the data was very similar to the control. That is the if we compare the LDH in control fish after 90 days, which was 0.427, and after administration of anthocyanin, it was 0.51. So we observed that after administration of anthocyanin, it was very near to the control fish. The protein profiling, next. If we see the SDS page of the protein profiling in the toxicated fish, we observe some special very dark high intensity bands of the stress related proteins, very overexpressed in these toxicated fish, having band size of 10.5, then 14, 22, 29. These were all very highly of the highly or overexpressed band size, and these are the toxicated proteins here. So coming to the last part of my research, that is the conclusion. The administration of anthocyanin to the toxicated fish species at an interval of 15 days resulted in reduction in toxicity induced by Takumi, that is the chemical name is Flubendiamide 20% WDG in Labeo rohita. And in this work, it has been shown that the liver and kidney being the detoxifying organs, the stress response is greater in these organs. And also we observed that after administration of anthocyanin, the toxicity level was very lowered in these soft tissues, and if we go for any further future extended programs on this administration of anthocyanin for much longer periods, then we just expect to see that the toxicity level will go further down, and it will very near to the close that control fish. And gill, liver, heart, kidney, muscle, spleen tissues are better applied in studying the isoenzymatic profiles for fish physiology, toxicology, and may also be used as a clue for toxicological studies. And also we see that the biochemical, hematological, enzymatic, and stress proteins to the normal condition to a great extent after administration of anthocyanin. And what is the future scope? So if we can if we restore the toxicated fish to the normal level, then we can attract much more people to go through the aquaculture and generate more ecological growth and lifestyle as a whole. And also bioavailability of the anthocyanins is very high, so it may be used to reduce the oxidative stress in various edible fish also. And the toxicity level will also go down, and anthocyanin containing the fish feed supplement maybe contribute to mitigate the loss of the fish population, thus increasing the proteinaceous food for the people or mankind. And at the same landscape or land resource, we can just produce some paddy crop also and some proteinaceous food also, and utilize the energy content from same landscape for the mankind. These are the some references which we use to study our research and observations also. And finally, thank you.